Okay, thank you very much, Dennis. Thank you very much also to David for putting this together. And it was a pleasure to work with you and Manishri and also Doelani at DSI and other colleagues at ICGB Cape Town. I really enjoy interacting with you. So I would like to start, I have 2 slides about ICGB. I'm sure there are many colleagues and friends who are connected, which I'm really happy that they are here taking part in this symposium. But just for those of you who don't know, ICGB, the International Center for Genetic Engineering and Biotechnology, is in fact an organization which constitutes of three centers, which we also call components. One is in Trieste and one in New Delhi. They were both created in 1987. And in 2007, we created one in Cape Town, South Africa. And together with three institutions, which we will make over, which constitute over 45 research groups, which do research in so many different fields. We also have now 66 member countries. We had one joining a couple of weeks ago. The last one is Angola. We're very happy to have them in the family. So we are together with 66 member countries. We are one family, and our mission is to interact and to promote biotechnology in our countries, especially in our 66 developing countries. So we have a lot of interactions and a lot of collaborations, and we are here for scientists in our member countries. In order to promote biotechnology and promote collaborations, we have several programs that they're all very well described on our website. But very quickly, we have cutting-edge research in our laboratories in Italy, India, and Cape Town. Italy is about 250 people. India is about twice the size. And Cape Town is about 50 people right now, but thanks to the South African government, is going to grow considerably in the next few years. And all these scientists are there for you, are there to interact with laboratories from our member countries. And we're all also here trying to do state-of-the-art science. We have our own, we have 3 major pillar programs. We have our own fellowships, which are exclusively reserved for scientists from our member countries, and we sponsor PhD. We have each component has its own PhD program, and also we have a postdoc program, and as well we host for short periods of time, both PhDs and postdocs. So please take a look at our fellowships program. We also are very interested in sponsoring meeting courses and workshops. We sponsor and co-organize or organize over 30 a year. And we also have one grant call a year where we give grants to laboratories in our member countries. This is a very popular and competitive program, and we're looking into ways to expand it. And also we have other services. But my recommendation is to look at our website, and please feel free to get in touch with any of the group leaders or any of our offices for any further information you might want. Okay. So coming to today, I have a group here in ICGB Trieste, and my interest has always been bacteria and bacteria that live in association with plants. And for many, many years, we've been interested in how bacteria signal and communicate with each other. And this has been quite a revolution for us, which really started over 15 years ago. The bacteria are not to be regarded as single cell. They are single cell organisms, but they do not want to live in solitary. They want to live as part of communities. And how do they do that? They communicate through small chemicals, which we also call languages, so that they can recognize their neighbors, whether they are friends, enemies, whether they are next of kin, whether they are distant. And they change their behavior according to who is around them and how many of them are around them. Now, I have no time. We're doing a lot of research. This is more fundamental research that we're doing, and we've been doing for many years. And this is a part of my group that we are very interested in this aspect, which in some way overlaps with what we're talking about today. Because since 5 or 6 years, I've been really interested, and we really also initiated research into the microbiome of plants and how this relates to plant health. I mean, we all know, just like, we humans have so many microbes that live with us. We have 10 times more microbial cells living with us than human cells. And they do so many important things. And it's the same is true for plants. And this is what, so this is now what we're interested in, understanding the microbiome in response to certain attacks by pathogens. And also, we are very interested in developing plant probiotics, and we are collaborating with several companies, but we really would like to focus also in the next years in projects with sorghum in Africa. So just as I'm the first speaker, just to highlight, I know, just to say something that we all know, but we should never forget, is that we have a very big challenge ahead of us, an enormous challenge ahead of us, which I don't know, probably in Africa and in other continents is much more evident than it is here in Europe or in North America, where most of the research that takes place involves biomedical aspects. But what we must never forget that by 2050, this is the FAO prediction, which is actually needs to be updated. We're going to be almost 10 billion people. And this is serious, because these 10 billion people, we need to eat. I mean, even today we have problems of a lot of people not getting enough food every day. Maybe it's more of a problem of distribution, but in 2050, it will also be a problem of producing enough food. And so the estimate is that we have to produce 60% more food. And guess what? From less cultivable land, because more land will be dedicated to humans living, and also we're dedicating more land to biofuels. There is climate change, which will create a lot of abiotic stress in some areas. So it's a very big challenge ahead. And guess what? We have to reduce the use of agrochemicals. We cannot afford to continue using the amount of fertilizers and pesticides that we're using today. It's just not possible. It's not possible for two reasons. It's not possible to produce so much of them every year. And secondly, we just cannot go on putting all this chemistry into our environment. Okay, this is a very important point, and that we all know, but it has to be really, really highlighted. So and I believe that solving this 60% will not be a eureka kind of, discovery. Oh, somebody will come up with an idea. This is it, guys. We do it like this, and we solved it. It's going to be a combination of a lot of different technologies, innovations, that together we will reach this incredible target. And one of these, in my view, it's going to be very important. It's going to be the plant microbiome. Now, just like every single organism on Earth and every single environment that we go on our planet, it's full of microbiology, right? And I have to go a little bit quick, so I apologize if I speak a little bit fast. But the plant microbiome, plants, just like all the animals, are full of microbiology, and there are a lot of studies coming up now about how many microbes there are, what type of microbes, all the networks involved. And the major drivers of the microbiome, what determines the microbiome, is the soil. Now, the soil is alive. The soil is the most biodiverse niche on the planet. 1 gram of soil contains 100 million bacteria. In 1 teaspoon of soil, there are more microorganisms than humans on Earth. Soil is where the food chain starts. So when we plant our crops into the soil, they have all this, they have a reservoir of the most biodiverse microbiology on Earth. Now, the host genotype also plays an important role. The plant will be there to recruit, to select the microbiome from the soil and feed it. And also there will be microbes which will already be present inside the seed. Okay, so these will be the vertically transmitted. So these are the three drivers that will determine a plant microbiome. And if we look at also the plant, we can divide it into compartments. Just like in humans, we have the oral microbiome or the skin microbiome. By far the most important is the gut microbiome. Just the plants is the same. Now, again, I don't have too much time. There is the seed. I already talked about. Endosphere is microbes that live inside plants, mostly in intercellular spaces. The phyllosphere is the surface, the aerial surface on surface of leaves, which this is a very important microbiome, because we know a lot of pathogens, especially fungi, attack, and also bacteria attack from the leaves. And the rhizosphere, this is the closest soil to the plant root. This is the gut of the plant. So this is a perfect parallelism with humans. This is this is by far the most important microbiome. Plants exude out into the rhizosphere. This is about up to 2 or 3 millimeters distance from the roots, the soil in this area is called the rhizosphere. And the plants exude out into the rhizosphere up to 20% of the carbon that they fix through photosynthesis. They do this in order to feed its microbiome and to keep it. And the microbial weight in the rhizosphere is 10 times higher than in the bulk soil. So in the bulk soil, we have the most biodiverse. It gets selected as it gets to the rhizosphere, and there's 10 times more microbes in the rhizosphere because of the food and elements that the plant provides. So selection, recruitment, and then feeding. And this is so important for its health because we know all the microbiomes, but especially the rhizosphere. It's so important. I apologize, I did not put the source of this picture, but it's from a review. I don't remember the authors. I apologize for this, but the rhizosphere microbiome, the gut of the plant, is so important for getting nutrients into the plant, for allowing roots to grow fast because a lot of microbes produce hormones, induce immunity, and then resistant to stresses, abiotic and biotic in these resistance, both abiotic and biotic. So understanding this plant microbiome, this rhizosphere microbiome, a lot of scientists are looking at this, and they're very complex. But it's, there are very important data coming out. There's a lot of it's dominated by 4 bacterial phyla. Again, I don't have time to go into, but it's very exciting research is being done in this microbiome. So what we started doing here in my group is, we looked at rice as our major. Is our model crop. We don't only work with rice, but rice is our model crop. And we started looking at several diseases of rice. And what we started looking at, sick plants, we go and look in the infection site, and we determine the complete microbial community, which is called the pathobiome. So all the microbial community in a disease is called a pathobiome. And in the same plant, in the same genotype, in the same area, in the same soil, but they're healthy, we determine in that same area the microbiome, and then we compare it. And we look at the differences, thinking about possible pathogen cooperator or possible beneficial bacteria. Because maybe the healthy plant, which is just maybe a few yards away, is healthy, maybe because it has a different, stronger, or more biodiverse, a good network. Whereas the plants that get sick, they were defeated. The pathogen made it and disrupted the microbiome, and it was able to take over. So we're doing these experiments from two different angles: pathogen cooperators in the microbiome and beneficial bacteria in the microbiome. Again, I apologize if I have to go a bit quick. We looked at 3 diseases of rice around the world: the sheath rot, which is caused by Pseudomonas fuscovaginae, which is a very important pathogen when rice is grown at high altitude. It's very common in Africa, Central Africa, Madagascar. And here, a good friend, PhD student, worked with me, Samson Muzonerimana, who is also connected to in this webinar. He's doing his PhD, just finished, looking at samples, infected and healthy, and doing some really nice comparison. He just very recently published a paper, if you want to look at that, if you're interested in that. Then we looked at a rice foot rot disease, which is important in Italy. Again, I apologize for going fast, but you can contact me for more details. And the rice leaf blight disease, which is incredibly important in Asia. Second most important disease of rice is caused by Xanthomonas oryzae, Pathomora oryzae. This bacteria enters through the leaves in the hydatodes and infects the vascular system of rice. And this has been a lot of work, collecting the samples, doing the analysis, and this is a lot of collaboration. We're doing all the paperwork, getting the samples, and I have to thank a lot of people here in the outstation of Burundi, Iri. Joseph Vigiliman is in charge of this outstation. He's been so helpful, and also institutes in Italy and in Vietnam, where we're doing all this work. But what we're seeing, if we look at some results, what we're seeing in the slide, looking at foot rot, for example, in Italy, caused by this Dickeatzea. What this graph means, this is what we've done is we take the infection site or the plant healthy site of the same zone in a healthy plant. We purify the total DNA and we do community sequencing. We amplify an area of 16S, and we get millions of amplicons, and we sequence them all. And then we analyze them. And if we look here at sick plant and healthy plants, over 30% of the amplicons in the sick plant is the pathogen. And if we look in healthy plants, we don't find the pathogen. So just as this, we look at the complete community in the sick and healthy plants, and all these colors means the presence and abundance of different bacteria. And you can see the patterns. I mean, we looked at hundreds of samples in different growing seasons in order to get statistical power, of course. Here I just show you, a representation of the results. But what we're seeing is that we see a significant, statistically very strong enrichment of certain bacterial genera in the sick plants. And what we think is that the pathogen will also be involved in enriching these guys, and maybe they have cooperative behavior. So the pathogen, we think, and we have evidence also in other studies using other plants, that the pathogen cooperates with some members of the microbiome, and they are involved in the disease process. The same we did, and again, we can select, doing analysis, which of the genera, bacterial genera and species are enriched in the disease, which members of the microbiome are enriched in the disease. And we're doing the same with Pseudomonas fuscovaginae in the disease of the sheath rot. These are all samples from Burundi, and we looked at disease plants, and we take the DNA from the disease plants, we do amplicon sequencing, and we find that in sick plants, we find the Pseudomonas, the pathogen. And again, we do the comparison. But in this case, we're looking at the ones that are really abundantly and differentially abundant in healthy plants. Okay, so again, just like I hope you have the animation on this correctly, but so just like you have with the foot rot, we're doing these comparative microbiome studies and pathobiome studies. We're identifying pathogen cooperators of the pathogen with one disease, and with the other, we're interested in looking putative antagonists of the disease to possibly use them as biocontrol agents. Okay. Again, apologies for going fast. So in the last 10 minutes or so of my talk, I want to present to you what we're doing with respect to very applied aspects of my research. And this is exciting for me and for my group because it's very new for us. We are doing a lot of science, a lot of training, trying to find new biological systems. But in this case, we're really thinking about applications. We at ICGB, as statute, we cannot initiate startups. Whereas, colleagues of ours in universities and institutes, it's now possible that you can do a startup. So what we would like to do, we initiated a platform, and we collaborate with companies, and we are collaborating with several companies to develop microbes as plant probiotics. I don't know if you see the whole, because here I have animation, but I'm not in an animation zone. I hope you see the whole slide. Anyway, so I already mentioned the importance of the rhizosphere microbiome in plant health. So I'm not going to go into detail on that. But, and a lot of the microbes there have a lot of advantages in using them as plant probiotics. They promote growth, they have low environmental impact, very easy to produce, well, relatively easy to produce, and you can have positive results for crop improvement. And what you see in this graph is that, as I was saying earlier on, that the modern agriculture cannot go on as it is going on now. And this is why using microbes as probiotics is a relatively exciting thing. It's not a new thing, but the new technologies, especially the ability to do microbiome studies, has generated a lot of new interest in using microbes as probiotics. And you can see here that the market for this biofertilizer and also biopesticides is increasing by 10% every year, and it's projected to do so, to continue to do so. So you can see between 2019 to 2025, it looks like it's going to double. This is, in billions of dollars. It's not much if you think about the biomedical pharma, but it's very important. It's increasing a lot. There's a lot of interest in this field. And there's a lot of different ways one can use them. One can use biological products. They can contain microbes. They can be organic compounds, extracts, compost, digestates. And microbes is what interests us. And you need to understand which is the microorganism, how much you need to use, which crops are you going to use it on. You have to have certification of agronomic efficacy, and so on and so forth. Whether it's a probiotic, a biofertilizer, a biostimulant, there are a lot of different names. So you have to specify. You have to specify and characterize what you're working with and what you want to bring to the market. There's a lot of advantages, as we will hear today. They're not pathogenic, they're not opportunistic. That's important. They have to be easy to grow. These are characteristics that microbes as probiotics must have. They have to be formulated. You have to be able to formulate them. This is important because you want to make them such a way, you want to grow them and keep them in such a way that the viability will not go down quickly. They must be resistant to industrial process. They have to be stable, and they have to be, and this is very important, they have to work in the field. And you can have different formulations, liquid, solid, or you can have innovative formulations. Okay, you can apply them in different ways. You can do seed coating, you can do foliar spray, you can also put it in the soil enrichment. Again, my apologies for going fast, but I want to make sure that I'm sticking to my time and not put under stress my colleagues who come next. So what do we do here in our platform Strains for Plants? We really focus on three aspects. We want to find the best strain. And now we're also focusing on finding consortia of strains, because we know that the microbiome is so complex, has a lot of different microorganisms that live together in a network. So we think that maybe using more than one strain is the way to go. We also want to make sure that the environmental conditions that we want to use it, this strain or consortia is very well adaptable. And that we also want to study, and we are studying, what is the effect of using this strain or consortium strain on the total microbiome? Is it changing it dramatically, or is it not? So what is the picture of the microbiology once we use our strain or consortia? And for this, we developed a pipeline in order to do this. And of course this can vary, can change, and can vary according to the company that we talk to. But we can isolate and select. Again, I have no time to go in all the details. We can characterize very well. We can do compatibility tests. We are interested to study which strains are happy together, which strains do not interfere with each other, which strains can colonize the rhizosphere very well or the endosphere very well together. We do the plant tests to make sure that these strain or consortia are able to persist and or have a beneficial effect on growth under different conditions. They can be optimal growth conditions, they can be stress conditions, abiotic or biotic stress conditions. And then we can provide a report on any of these aspects. And you can see here, some of the pictures of the things we do. We isolate and characterize. We do compatibility tests. We use microbiome analysis. We use growing, also more traditional. We're not only using NGS, we're also using traditional ways to make sure they are compatible. We do a lot of different plant tests, and we all do this with statistical strength, and then we provide a final report. This is it. This is what I want to, this is the overview I want to give of my lab. I mean, my lab, this slide is not very fair because these are the people that are in my lab or coming in the lab at the moment. We have 2 PhD students working, and we have one, Shandan, is now, he should have been here in October, poor guy, and we are doing our best with the COVID pandemic to get him here. Now it looks like he's going to come in early January. He's going to join our team. We are looking forward to host him. We're also going to have a postdoc, 2 postdocs, and lab manager and research associate. I mentioned Samson, who just left our lab. Had a really good time having him here at PhD. I have a lot of collaborators, but I've had a lot of people that come and go, and a lot of other scientists, which has been a pleasure collaborating through the years. These are, funding is a very important, funding and collaborations are very important. Here it's funding and also collaborations that we have with institutes or funding agencies. And these are the companies that we are collaborating at the moment. but we are talking to also several other companies. On a good day, I look like this. This is my email. So again, I had to give an overview today. I cannot give a lot of our, many of our details. So please, I'm really happy to hear from you in my email with any comments or if you have any idea of any collaboration. Doesn't have to be, it can also be, you can contact me if we see we can collaborate on some science, or if you're interested to do some practical collaborations. We'd be very, very happy to hear from you. Okay, thank you very much.
Good. Thank you very much, Vittorio, and thank you very much for keeping the time. You spent exactly 25 minutes.
I had to go a little quick. I'm sorry about that. Yeah.
So thank you very much. So we have 5 minutes for questions, and maybe I will start with some of the questions that have been posed by the participants. And one of the questions is, Vittorio reads, how best is genetic stability attained in biofertilizers to reduce risk of mutations which may become problematic in the long run? So how do you ensure that those things do not mutate and become a problem?
I mean, it's a very big question. I mean, we are putting, with using plant probiotics, obviously you're enriching, you're augmenting your microbe into let's say the rhizosphere. So you're creating an imbalance, and this is what we're looking at. We're trying to understand, by putting these probiotics, yes, what is the effect on the total microbiome. I think it's basically we're using what I want to make very clear. We're using wild type bacteria, and this field of research applications is not intending to genetically modify any of the microorganisms that we're going to use.
Yes, yes.
So they are in nature, and we know that in nature mutations happen and things change, and this you cannot stop. This you cannot. I am personally not too worried about it. There could be changes, but these happens in nature all the time. So we cannot make them genetically more stable because we're not going to modify them. And yes, there will be changes, but we're using harmless beneficial bacteria, so I'm not too concerned about that. I hope I've answered the question.
Yes. But Amoyo, I hope your question's been addressed. But one other quick question, Vittorio.
Tell me, Dennis.
I'm wondering, I mean, are there specific microbes that you have come up with recently in your research projects that could have potential applications, say, in Africa? And if so, are there any initiatives that have been put in place to try and see if these products could eventually be assessed in Africa for purposes of addressing issues that we're experiencing this end?
Well, Dennis, thank you very much for this question, because most of our work so far has been, and we're collaborating with companies we are collaborating with companies in China and also in the United States and in Europe. But what we want to do now, and we're very excited to do now, is that we're starting now in my lab. We're going to focus on sorghum, and we want to, which is the African cereal, right? And we want to work very closely with sorghum, in some African countries, in order to isolate consortia of specific bacteria that we have in mind in order to produce products for sorghum in Africa. So we initiated this program right now, and this is why I'm so excited to take part in this symposium and to look for I'm in touch with the CSIR in Pretoria, that we have some ideas, and we want to really work in the sorghum in Africa with collaborators in Africa. This is my aim, my biggest aim in the future.
Oh, okay. Thank you.
Thank you.
Do you have any other question, David?
Yeah, so I just, thanks a lot, Vittorio, very interesting. And I'm a sorghum person myself, right? So, and in the SADC region, we do have a sorghum focal point, which is I don't know if you know this, at Matopos in Zimbabwe. So they're the small grains people.
Oh, great. Yeah.
And, yeah. So it's really sorghum, and maybe also look at the millets. So
Okay,
finger millets.
Yeah, yeah. Okay.
Yeah, because those are climate smart crops, and we really need to get people going back to those grains because of climate variability and things like that. Yeah. But
so anybody who's interested in this, please contact me. Yeah.
Yeah, thank you very much. Yes, yeah, there are a couple of comments, people asking for contact. So we'll try and share those. There are a number of people who are interested in collaborations.
Very
and
good.
as you said, yes, if you have any comments or questions, please feel free to post them in the chat box, and we'll try and ensure that they get answered.
And I've also given my email. I've also given my email at the end, so anybody can have to contact me. Yeah.
Okay. So I'm looking at the time, and unless there's any other question, there's still time. You can still post your questions on the chat box, and we'll try to get as many of them as possible addressed.